there, and welcome to another edition of Catalysts, a platform for the scientific community to connect, discover, and transform. This is your host, Kedar Aris, and today I have the pleasure to connect with Dr. Tara Deans, Assistant Professor of Bioengineering at the University of Utah. Dr. Dean's research is in the field of applied synthetic biology. Her lab focuses on developing new technologies to regulate gene expression in mammalian cells with applications in tissue engineering and regenerative medicine. So, what is synthetic biology? It is a relatively new field that builds on the knowledge base of genetic engineering and cell and molecular biology, with focus on re-engineering of naturally occurring biological systems or designing new biological entities such as enzymes, genetic circuits, and cells to solve specific problems. Example, control the spread of cancer or regenerate damaged bone tissue or cartilage, etc. Dr. Dean's lab is currently focused on combining synthetic biology and materials science to engineer cells as therapeutic entities, used engineered cells to manipulate the extracellular matrix to support differentiating stem cells, and engineering biologically inspired microenvironments to better understand the cellular mechanisms in both healthy and diseased tissue. Dr. Deans has won multiple awards for her pioneering work in synthetic biology. And as she charts a stellar academic career, she exemplifies the spirit of catalysts, connecting with people and building lasting relationships and discovering the benefits of collaborative research, which is helping to transform her career as well as the careers of others. Dr. Deans, thank you for joining us on the Catalyst podcast.
Thank you for having me.
My first question was, you did your PhD in biomedical engineering
Yes.
at Boston University under Dr. Jim Collins. And from there, you ended up doing your postdoc at Johns Hopkins with
Yes.
Dr. Jennifer Elisif.
Yes.
So can you walk us through that transition from
Sure.
grad school into...
Sure. So when I did my graduate work in a field called synthetic biology, and that was really during when it first started becoming a field. I actually was at SB 1.0. And really what the field does is it reprograms cells to behave in predictable and controllable ways. And I was one of the first people to work with mammalian cells and building these genetic circuits in them. They're just composed of a bunch of different modules. And we use the different modules to control gene expression. And so I built a genetic switch in mammalian cells, and towards the end of my PhD, I became very interested in applying this new technology for human health. So I wasn't quite sure how to do that. And so I started looking into the options and opportunities, and I thought the most realistic way to transition was to actually look into biomaterials. And I was also very interested in stem cells. And so Jennifer Elisif's lab at Johns Hopkins is actually very well known for the materials that she has been working on for 10, 12 years, and also with stem cells. And so it really was a perfect match. And she was also very open and willing to have somebody like me come in and sort of shake things up in the materials world. So. And
did you directly approach Dr. Elisif, or did you go through Dr. Jim Collins to kind of network?
No, at that point, I was a big girl, so I was very much encouraged to do things on my own. So I contacted Jennifer, and it took a couple of tries. She's very busy, as most very well-established and successful PIs are. And it took a couple tries, and I was able to finally get in contact with her. And she said, sure, come on down for an interview. And I did, and I met everybody in the lab. And and then she offered me a position. So.
Then during your postdoc, was it a 2-year or a 3-year postdoc?
It was like 4 years.
4 years.
So, my time it was a little extended because I had children. I had 2 children at graduate school and 1 child in my postdoc. And so my time frame, I think, is very abnormal for most biomedical engineers because I just had other things happening in my life that prevented me from going to the lab as frequently as I did before children.
And during that postdoc period, I often hear professors saying that is the time when you have to be proactive, when you have to be aggressive. You gotta look for grants, you gotta bring up ideas to your PI, and you gotta look for opportunities. And I imagine that's a pretty interesting, exciting, but still anxiety-inducing time.
It is. The word aggressive, I don't know if aggressive is the right word. I think being assertive with your ideas, but also really listening. I think listening is it's an undervalued trait that people I think often forget to do. And because there is so much that is said and done if you just listen. And so obviously talking to your PI is important, but also talking to other people in the field is equally as important. And I was able to get funding right away in my postdoc. I was really lucky to be able to do that. So that did give me the freedom to do a lot of what I wanted to do during my postdoc. But it's also about finding that right match with somebody that is open to your ideas, and also trying to integrate those ideas with what the lab actually does. So that's also important. But yes, talking is important, and trying to find, even just getting experience of grant writing, because when you're on your own in an assistant professor position, it's very different. You're much more isolated. You don't have someone's door to go knock on to say, can you read this? So trying to get that experience during your postdoc is important. Even in grad school. Some students also get it in grad school too. But the more experience you get, the better.
Got it. Was it a K-99 funding, or was it a
I had a Maryland stem cell grant to apply synthetic biology to stem cells and materials.
Got it. And then from postdoc transitioning into University of Utah, how did that process go?
Well, it actually involved rallying a lot of support around me. I have a lot of really amazing mentors in my life. I'm very, very fortunate, and so both Jim and Jennifer, but also a lot of other people. There are people here that I knew. John White came from BU, and it's just really about letting people know that you're on the market. BMES has a great program where it's Meet the Faculty candidate, and it's actually the evening before the whole meeting starts. So that's an opportunity to just say, hey, I'm going to be on the market soon. And that actually is coupled with, I believe it's a chairs meeting, a department chairs meeting. And so usually after the meeting or before, I think it sort of toggles between meetings, all the chairs either go before to Meet the Faculty and then go to their meeting, or they go to the meeting and then go visit the faculty that will be on the market. So it's a nice way also to for them to see who's going to be on the market in our field, and then also for you to really get an idea of what's out there, of the opportunities that are coming out, and because sometimes it takes departments a little bit of time to post their ad.
Right.
But it's coming, and then you can kind of, do your homework on the department before, and then you can have your application in right away.
What are the requirements that they look for? Do you have to have your own funding? Do you have to have your established?
Well, I didn't. Going into I have funding for my postdoc, but I didn't have funding. I'm all about startup, and I think that my impression, I've been here. It's been one full year now.
Right.
Of the offers that I had, and the places that I visited, the one thing that every place had in common was they were really looking for the best person. And some places do have an agenda where they need somebody to teach a certain class, or they need someone who does a certain kind of research. And so those you'll be screened, and you won't interview for those unless there is that match.
Right.
But in terms of getting yourself ready for the interviews and all that, you have to write your teaching statement.
Mm-hmm.
You have to write your research that you want to bring to the university. And so my advice in those documents is, for the teaching, really go through the curriculum at every single university and figure out what they teach, figure out where their holes are, and if someone is leaving or has recently left, see if you can teach something in that area, because generally they're looking to for someone to pick up those classes if someone has left recently. And so but it's also important to talk about the class that you want to develop at the university. So generally you'll probably teach a core course that's already available to be taught, and then they'll ask you to develop a class, bring in your expertise, and integrate it with the current curriculum. And so those are the two sort of aspects that are important in the teaching statement.
And the funding varies from university to university.
Yeah, I honestly didn't have, that was not an issue for me. Okay. I didn't have a K-99. I didn't have a Career at the Interface of Science Award. I didn't have any of those, and that didn't seem to matter.
Interesting. The next kind of segue I wanted to cover was the research, and you mentioned synthetic biology. Seems there isn't a hard established definition of what synthetic biology means. Everyone seems to have their own definition.
Right.
So can you kind of just share what your definition of synthetic biology is?
So my definition is the ability to reprogram cells to behave in controllable and predictable ways. And also that behavior is also related to dynamic behaviors, being able to turn gene expression on, off, regulate its expression. If you think of electronic circuits and all in the different behaviors that you can have a circuit board do, you can essentially do a lot of the Boolean logic in a cell by using tools in synthetic biology.
Got it. So it's like genetic inducers or repressors.
Right, and you can build AND gates, OR gates,
NAND
gates, all of that, yeah.
Interesting. So from synthetic biology, you did mention how you wanted to kind of expand into the translational research realm, and that's where the biomaterials amalgam kind of it was a natural fit or a match. Was that something you already had in mind when you explored postdoc opportunities, or was that something that kind of fleshed out in more detail?
Well, I think both. I think I had never worked with biomaterials before, and so I had this very romantic idea that I would go and do my postdoc and I would just figure out how to get these cells into people, because you can't just, it's very different going from growing cells in 2D to putting them into a person. And but then when I started my postdoc, I started to realize that it wasn't going to be as straightforward as I was hoping. And so I think the materials, big picture materials idea was really in my PhD, because I've always wanted to use these cells for therapeutic applications. And so my biggest question was, well, how on earth would I get these cells into people? But then it was during my postdoc when I started to really learn to appreciate, both from other postdocs, from students, and certainly from Jennifer, how complicated this can be, but also how exciting it can be, because you can, especially working with something like a PEG hydrogel, you can go in and work with the different chemistries to get various properties in the gels, and in the different materials. And so that was fun. That was also, obviously very much related to the lab that I was in. And so, and I also worked with a chemist who was able to help put some of these inducer molecules on different materials to induce gene expression as well. So it was sort of both, an early idea that matured with expertise around me. So,
and collaboration probably played a big role into
definitely
shaping that research. Which kind of brings me to the next question, in terms of identifying research ideas to kind of continue and expand on your research.
Right. Yeah. So that's really that's the fun part. Well, there are two fun parts for me. The first part is coming up with the ideas, and that is I read a ton. In the scientific literature. I also talk a lot, and so I'm constantly talking with people about their work, my work, and and this sort of goes into the collaborations, and really there's so much to be done. There's so much that is undiscovered, and really it's about what do two people have or three people have that can bring something really unique to the table to try to go after some of these much harder questions in human health or disease and things. So, in terms of the actual development of the ideas that comes from reading the current literature, coming up with some really tough questions, or even just being able to identify, in the writing where you can sort of see where the gaps are and the bigger questions are and things, and then when I start writing the grants or start writing some of this up to either entice one of these people that I've talked to that maybe we should work together, or even just to write a grant, I really go after more of an investigative, what is known, what is unknown, and so there I actually read a lot of crime novels. And so because for me it's a lot about just solving a mystery, and it's a lot of you have some fact, so you have some mystery identified, and you have to figure out what is the information that what is the usual suspect, and then always it's never what you think it is. It's never the protein you thought it was. It's never the material you thought you should use. It's very similar to that.
But it comes to collaborations. What is your philosophy like? Is it something where you actively seek out new collaborations, or is it something where people come to you and then if there is similar match or interest?
I think it's both. I've had experiences where people have come to me and said, I'm trying really hard to try to solve this problem, and I think that the tools that you have and the tools that I have, if we can interface them somehow, then we can really start to go after this this long question that's been in the field. But also there's the situation too where I have friends that we just want to work together,
right? And
we try to come up with ideas of ways that we can collaborate and work together.
So the next kind of step was, okay, you have identified the research, and you have identified resources in terms of collaborators. But in the end, the success depends on how much funding you can get, and which is where the whole process of grant writing and successful grants become a key factor. So my question was, how much time do you spend writing grants, and what does it take to write a successful grant?
Well, so I just started. So I have the only successful grant I've written is my Maryland stem cell grant. But and so I am starting to apply for grants now, and so hopefully you can ask me this spring, but about the successful ones. But it's funny because no one. This job, there was one big surprise for me in this job, and that is I never wanted a desk job, and I always imagined myself as this little old lady with bench space, always doing experiments, but that's what I love to do. I love being in the lab. I love being a part of it. And so I try to get in the lab as much as I can. It's still early for me, so I do have that time. But it's the grant writing. It's a lot, but it's getting easier now that I have one big one written, because then there's a whole body of work that I can just draw from my writing from, and so it's much harder for me anyway to look at a blank computer screen with that cursor blinking, but it's much easier to edit something, and so just getting something down initially and then going back and editing it helps with the process. But yeah, I feel like I spend a lot of time right now, but I think I'm supposed to. I think that's normal for somebody in their first year.
How do you identify which grants to apply for, or is that
Well, so
trick to it?
I have a lot of help from my chair,
Okay.
who encourages me to apply for things. I have, I have other, colleagues that send me different announcements, so, and I look at, I look for different things, and all of it together. It's recommendations from colleagues, from my chair, from my friends, all of it together. It's a huge stack of grants to apply for, and yeah, and that's actually helpful too, because then I can sort of sift through to see what I qualify for, or what I might have a chance at getting,
Right.
based on my work.
The last piece of the puzzle in the whole lab is identifying the right people to hire.
That's so important.
Exactly. So the question was, what do you look for when you're hiring grad students or postdocs?
I guess two things. The first is interest, and the second is motivation. I don't necessarily think that the student with the highest grades equals the most successful graduate student. I think it's often just someone who has that fire in their belly, and they just want to. They're just so excited about their project, and they're so excited about their work that they come in and they get things done. And it's I have a really wonderful group now, and it's everyone's working very well together, and I've been very, very lucky. So
during your time as a researcher, have you had any setbacks or maybe failed experiments?
Oh yeah, who doesn't? Yes, there. I feel like grad school and even my postdoc, it's just a roller coaster ride. You have, peaks and valleys. You have, it's just a wild ride, and I think it's you just, and honestly, just getting through it all, it's really always been my interest in the science and my love for what I'm doing, because there are times when it gets very hard because nothing's working. You feel like, what's so interesting is, you go through life reading textbooks. You go through life reading, until you reach graduate school where, science has laws, math has laws, physics has laws. Someone else figured them all out. And I always just had this assumption that all of these brilliant people, previous to us that helped write these textbooks just knew what they were doing, and they wrote these laws. And it wasn't until I got to graduate school that I realized a huge part of learning was failure. And and a huge part of my success was built off of working through a lot of really tough problems. And if I wasn't persistent, and if I wasn't maybe a little stubborn with just getting through it so that I can just finish what I started, it's tough. I think everybody has a plan B in graduate school. So I think everybody at some point has a plan B.
Got it. So what I'm hearing is failures are a part of life as a researcher, and the challenge is not only to see it for what it is, but use it as a stepping stone to get to the...
Well, and also just don't, you cannot beat yourself up constantly. It's counterproductive, because anyone entering graduate school or a postdoc thinking that they will never fail is just setting themselves up for depression. And it's, it happens to everybody, and I don't think that you really realize that it's totally normal until you're on your way out. And you look back and you're like, wow, that was really tough, but I made it. And then you start talking to your friends that are recently out or on their way out of graduate school, and there's this resonating theme that, man, year 3 or year 4 or whatever year was really rough. I didn't think I'd ever get through it. And that's why I actually can't emphasize enough to put positive role models around you. And and it doesn't have to be your advisor. It's okay to have other people in the department, other people in other departments. And it's really smart to put those types of people around you, so that when it's time to help you get back on
pick me up.
Yeah, I mean, they can rally around you and help. That's really important.
Would you attribute that as one of your keys to being successful? Having the right mentors?
Well, it's a lot of things, actually. It's the unwavering support from my husband and my family. I could never do this on a daily basis if I didn't have that kind of cheerleading regularly. And then there's just the handful of people that have been there. When I've walked into various offices with, I don't think I can do this, and they've talked me out of it. For me, my success is their success, and I think that people, when I talk to them now, that I've known, like Jim Collins, for example, I just saw him at BMES, and it's just so comfortable. It's just. He's just someone that's been in my life for so long at this point that it's just nice. I know that I can call him for anything, and I have lots and lots of mentors like that.
Taking slightly different tack, your research area would be synthetic biology, biomaterials, tissue engineering, and cell-based therapies.
Yes.
How do you see this field evolving in the next 5 to 7 years? What are some of the exciting things that are coming down the pipe, and what are some of the challenges you see?
Yeah, so I believe that synthetic biology will change how medicine is practiced. I believe that having tight control of cells, to be able to navigate where they go, to be able to have them seek out damaged and diseased tissue, and then once arriving, either delivering a bolus of a pharmaceutic or slowly releasing some kind of therapeutic molecule. I believe that will be the future of how many problems are treated, health related problems are treated. There are a lot of challenges with that. Obviously, recombinant DNA in cells. There's the FDA. I don't think anybody in this career wants anything other than to cure or to have that next step to either prolonging life or and things. And so I do think that coming up with more novel ways of regulating gene expression that either include or not include DNA will be probably the next step. Yeah, whether it's RNA switches, which has been a big deal lately, and things like that. So I mean, synthetic biology is, even like the paper-based synthetic biology. That's the newest thing off the press. That's been huge, and it's all about diagnostics and being able to detect scary pathogens like Ebola and things. So and these are oftentimes synthetic biology is being used now for for a more sensitive diagnostic or a better way to get a cell to go into a specific location, and then like going back to our earlier conversation, is adding in this Boolean logic, the AND gates, the OR gates. It's like the cell will arrive and say, know that it's there because it says I'm here in the brain, and I see inflammation, I will turn on. And so it's that kind of sort of program cell that's what we're building here, is that kind of cell.
You're just starting your assistant
yes.
professor track, but 15 years from now, 20 years from now, as you look back, what would you like your legacy to be in terms of the way your peers see you, the way your students
Right.
see you?
Well, I think it's twofold, to be honest. I would like my students, I would like part of my legacy to be my students and the success of my students and postdocs. We have a lot of fun here. We do a lot of great things, but I would like them to, a part of my legacy to be them. But the other is the technology that we build here, and that it really helps people, whether it's a diagnostic or a therapeutic. That's my hope. Is in 20 years, when you come back, we can talk about that.
Definitely. Well, Dr. Deans, this has been a fascinating conversation. Thank you for taking the time to join us.
Thank you very much.
If you liked what you have heard, I encourage you to share this episode with a friend. I'd also love to hear your thoughts on how you are striving to be a catalyst. So I invite you to leave a comment on the Catalyst's website. Thank you for listening, and until next time, connect, discover, transform.